Excretory product and their elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and iron like sodium, potassium, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with special emphasis on common nitrogenous wastes. Ammonia, urea, and uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is ammoniotolism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotolic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surfaces or through gill surfaces. In fish, as ammonium ion kidney do not play any significant role in its removal. Terrestrial adaptation necessitated the production of a lesser toxic nitrogenous waste like urea and uric acid for conversion of water. Mammals, many terrestrial amphibians, and marine fishes mainly excrete urea and are called uretilic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures in most of the invertebrates. These structures are simple tubular forms, whereas vertebrates have complex tubular organ called kidney. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structure in platyhelminthes flatworm, example planaria, rotifers, some annelids, and the cephalochordate amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, i.e. osmoregulation. Nephridia are the tubular excretory structure of earthworms and other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malphizian tubules are the excretory structures of most of the insects, including cockroaches. Malphizian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal gland or green gland perform the excretory function in crustaceans like prawn. Human excretory system. In humans, the excretory system consists of a pair of kidneys, one pair of ureters, a urinary bladder, and a urethra. Figure 19.1. Kidney are reddish brown bean-shaped structure situated between the level of last thoracic and third lumbar vertebra close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Toward the center of the inner concave surface of the kidney is a notch called hilum through which ureter, blood vessel, and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis with projection called calyces. The outer layer of kidney is a tough capsule. Inside the kidney, there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids projecting into the calyces, seeing a calyx. The cortex extends in between the medullary pyramids as a renal column called column of Bertini. Each kidney has nearly 1 million complex tubular structure called nephron, which are the functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of renal artery. Blood from the glomerulus is carried away from by an efferent arteriole. The renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus, along with Bowman's capsule, is called the Malpighian body or renal capsule. Corpuscles. The tubule continues further to form a highly coiled network, proximal convoluted tubule. PCT, a hairpin shaped Henle's loop, is the next part of the tubule which has a descending and an ascending limb. The ascending limb continue as another highly coiled tubular region called distal convoluted tubule DCT, and the DCTs of many nephrons open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscles, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephron. In some of the nephron, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephron. The efferent arteriole emerging from the glomerulus form a fine capillary network around the renal tubule called the peritubular capillaries. A minute vessel of the network runs parallel to a Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephron. Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion that takes place in different part of the nephron. The first step in the urine formation is the filtration of blood, which is carried out by the glomerulus and is called glomerular filtration. On an average, 1100 to 1200 milliliter of blood is filtered by the kidney per minute, which constitute roughly one-fifth of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, i.e., the endothelium of glomerular blood vessel, the epithelium of Bowman's capsule, and a basement membrane between these two layers. The epithelial cell of Bowman's capsule, called podocyte, are arranged in an intricate manner so as to leave some minute spaces called filtration slit or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass into the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The amount of filtrate formed by the kidney per minute is called glomerular filtration rate (GFR). GFR in a healthy individual is approximately 125 milliliter per minute, i.e. 180 liter per day. The kidney have built-in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus JGA. JGA is a special sensitive region formed by cellular modifications in the distal convoluted tubule and the afferent arteriole at the location of their contact. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of the volume of the filtrate formed per day, 180 liter per day, with that of the urine release, 1.5 liters, suggests that nearly 99 percent of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cell in different segments of the nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acids, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by the passive transport. Reabsorption of water also occurs passively in the initial segment of the nephron, figure 19.5. During urine formation, the tubular cell secretes substances like hydrogen, potassium, and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid-base balance of body fluid. Function of the tubules. Proximal convoluted tubule (PCT) is lined by simple cuboidal brush border epithelium which increases the surface area for reabsorption. Nearly all of the essential nutrient and 70 to 80% of the electrolytes and water are reabsorbed by the segment. PCT also helps to maintain the pH and ionic balance of the body fluid by selective secretion of hydrogen ions and ammonia into the filtrate and by absorption of HCO3 from it. Hence, loop reabsorption is minimum in its ascending loop. However, this region plays a significant role in the maintenance of high osmolality of medullary interstitial fluid. The descending limb of loop of Henle is permeable of water but almost impermeable to electrolyte. This concentrates the filtrate as it moves down. The ascending lip is impermeable to water but allows transport of electrolyte activity or passively. Therefore, as the concentrated filtrate pass upward, it gets diluted due to the passage of electrolyte to the medullary fluid. Distal cannulated tubule DCT conditional reabsorption of sodium and water takes place in this segment. DCT is also capable of reabsorption of HCO3 and selective secretion of hydrogen and potassium ion and NH3 to maintain the pH and sodium potassium balance in blood. Collecting duct. This long duct extends from the cortex of kidney to the inner part of the medulla. Large amount of water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of a small amount of urea into the medullary interstitium to keep up the osmolality. It also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of hydrogen and potassium ions, figure 19.5. Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle loop and vasa recta play a significant role in this. The full flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus forms a countercurrent. The flow of blood through the two limbs of vasa recta is also in a countercurrent pattern. The proximity between the Henle loop and vasa recta as well as the countercurrent in them help in maintaining and increasing osmolality toward the inner medullary interstitium, i.e. from 300 osmoliter in the cortex to about 1200 osmoliter in the inner medulla. This gradient is mainly caused by the NaCl and urea. NaCl is transported by the ascending limbs of Henle's loop, which is exchanged with the descending leaf of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amount of urea enter the thin segment of the ascending leaf of Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of substances facilitated by the spatial arrangement of Henle's loop and vasa recta is called the countercurrent mechanism, figure 19.6. This mechanism helps to maintain a concentration gradient. In the medullary interstitium, presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby Concentrating the filtrate urine, human kidney can produce urine nearly 4 times concentrated than the initial filtrate formed. Regulation of kidney function. The function of the kidney is efficiently monitored and regulated by the hormonal feedback mechanism involving the hypothalamus, JGA, and to a certain extent the heart. Osmoreceptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate these receptors, which stimulate the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitates water reabsorption from the later part of the tubule, thereby preventing diuresis. An increase in body fluid volume can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictory effect on blood vessel. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The JGA play a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure GFR, can activate the JG cells to release renin, which convert angiotensin in blood to angiotensin first and further to angiotensin second. Angiotensin second, being a powerful vasoconstrictor, increase the glomerular blood pressure and thereby GFR. Angiotensin second also activates the adrenal cortex to release aldosterone. Aldosterone causes the reabsorption of sodium and water from the distal part of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in blood flow to the atria of the heart can cause the release of atrial natriuretic factor ANF. ANF can cause dilation of blood vessels and thereby decrease the blood pressure. ANF mechanism therefore act as a check on the renin-angiotensin mechanism. Micturition. Urine found by the nephron is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system CNS. This signal is initiated by the stretching of the urinary bladder as it gets filled with urine. In response, the stretch receptor on the walls of the bladder send signal to the CNS. The CNS passes on the motor message to initiate the concentration of smooth muscles of the bladder and stimul- uh, simultaneous relaxation of the urethral sphincter, causing the release of urine. The process of release of urine is called micturition. And the neural mechanism causing it is called the micturition reflex. An adult human excrete on an average 1 to 1.5 liter of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic, pH 6.0, and has a characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristic of urine. Analysis of the urine help in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, glycosuria, and ketone bodies, ketonuria in urine are indicative of diabetes mellitus. Role of other organ in excretion. Other than the kidney, lungs, liver, and skin also help in the elimination of excretory waste. Our lung remove large amount of carbon dioxide, approximately 200 milliliter per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secrete the bile-containing substances like bilirubin. Biliverdin, cholesterol, degraded sterol hormone, vitamins, and drugs. Most of these substances ultimately pass out along with digestive waste. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat gland is a watery fluid containing NaCl, a small amount of urea, lactic acid, etc. Through the primary function of sweat is to facilitate a cooling effect on the body surface. It also helps in the removal of some of the waste mentioned above. Sebaceous gland eliminates certain substances like sterol, hydrocarbon, and waxes through sebum. This secretion provides a protective oily covering for the skin. Do you know that a small amount of nitrogenous waste could be eliminated through saliva too? Disorder of the excretory system. Malfunctioning of kidney can lead to the accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from the convenient artery is pulled into a dialysing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid dialysing fluid having the same composition as that of a plasma except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of a molecule based on the concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The cleared blood is pumped back to the body through a vein after adding antiheparin to it. This method is a boon for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of the acute renal failure, kidney failure. A functioning kidney is used in the transplantation from a donor, preferably a close relative, to minimize its chances of rejection by the immune system of the host. Modern clinic procedures have increased the success rate of such a complicated technique. Renal calculi. Stone or insoluble mass of crystallized salt, oxalates, etc., found within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney. Summary. Many nitrogen containing substances, the ions, the carbon dioxide, water, etc., that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretory vary among animals mainly depending on the habitat, availability of water. Ammonia, urea, and uric acid are the major nitrogenous waste excreted. Protonephridia, nephridia, malphizian tubules, green gland, and the kidney are the common excretory organ in animals. They not only eliminate nitrogenous waste but also help in the maintenance of the ionic and acid-base balance of the body fluid. In humans, the excretory system consists of one pair of kidney, a pair of urethra, a urinary bladder, and a urethra. Each kidney has over a million tubular structures called nephrons. Nephron is the functional unit of kidney and has two portions, glomerulus and renal tubule. Glomerulus is a tuft of capillaries formed from afferent arterioles, fine branches of renal artery. The renal tubule starts with a double-walled Bowman's capsule and is further differentiated into a proximal convoluted tubule (PCT). Henley's loop HL and distal cannulated tubule DCT. The DCT is of many nephrons joined to a common collecting duct, many of which ultimately open into the renal pelvis through the medullary pyramids. The Bowman's capsule encloses the glomerulus to form malphizian or renal corpuscles. Urine formations involve three main processes: filtration, reabsorption, and secretion. Filtration is a non-selective process performed by a glomerulus using the Glomerular capillary blood pressure, about 1200 milliliter of blood is filtered by the glomerulus per minute to form 125 milliliter of a filtrate in the Bowman's capsule per minute. GFR. GGA, a specialized portion of the nephron, plays a significant role in the regulation of GFR. Nearly 99% of reabsorption of the filtrate takes place through different parts of the nephron. PCT is the major site of reabsorption and selective secretion. HL primarily help to maintain osmolar gradient 300 osmolar liter to 1200 osmoliter within the kidney. Interstitium, DCT, and collecting duct allow extensive reabsorption of water and certain electrolyte which help in osmoregulation. Hk and NH3 could be secreted into the filtrate by the tubules to maintain the ionic balance and pH of body fluids. A countercurrent mechanism operates between the two limbs of the loop of Henle and those of vasa recta, capillary parallel to Henle loop. The filtrate gets concentrated as it moves down the descending limb but is diluted by the ascending limb. Electrolytes and urea are retained in the interstimum by the arrangement DCT and collecting duct. Concentrate the filtrate about 4 times, i.e. from 300 osmo per liter to 1200 osmoliter, an excellent mechanism of conversion of water. Urine is stored in the urinary bladder till a voluntary signal from CNS carries out its release through urethra, i.e. micturition. Skin, lung, and liver also assist in excretion. Thank you.